Welcome to lecture 2 of lipids. So in this lecture, let's start with saponification and soap. So what is saponification? So it is the reaction of fat with a strong base such as NaOH in the presence of heat. What does it do is it split triacyl glycerols into glycerol and sodium salts of the fatty acids. So this is the main process for creating soap. So soaps are basically salts of fatty acids. So this process gives solid soaps that can be molded into different shapes when mixed with NaOH. Now it gives it softer and liquid soaps when you mix it with KOH. So what is happening here? When you add fat or oil to a strong base in the presence of heat, it splits it into glycerol and the salts of fatty acids. So the glycerol and the salts of fatty acids split and form. So the one that splits into the salt of fatty acids is what we call the soap. So that's why the name saponification is basically creation of soap. So we discussed 4 different reactions. First one is esterification, where you mix a carboxylic acid and an alcohol in presence of acid and heat. It creates the ester and water. Hydrogenation is the process where we take an alkene and add hydrogen to create an alkane in presence of a metal. Hydrolysis is the process of adding ester and water together in the presence of an acid and heat creates carboxylic acid and alcohol. Hydrolysis is the opposite of esterification. Saponification is where we add ester and sodium hydroxide to create a sodium salt of carboxylic acid and alcohol. So these are the reactions that we have. So pause the video right here and try to write the equation for the catalyzed of the enzyme lipase that hydrolyzes glyceryl trilaurate during the digestion process. Next, let's go to the next main topic, phospholipids. So what are phospholipids? Phospholipids are a family of lipids that are similar in structure to triacyl glycerols, but they include glycerophospholipids and sphingomyelin. So you have a glycerophospholipid includes a glycerol and 2 fatty acids attached to a phosphate group and an amino alcohol. So glycerophospholipids contain 2 fatty acids that form an ester bond with the first and the second hydroxyl groups of glycerol, and a hydroxyl group that forms an ester with a phosphoric acid, which forms another phosphoester bond with an amino alcohol. So next, let's talk about sphingomyelin. So sphingomyelin contains sphingosine as the instead of glycerol here. So it contains a fatty acid and a phosphate and an amino alcohol. So it just contains 1 fatty acid and a phosphate and an amino alcohol. So what are amino alcohols? Amino alcohols are generally that are found in glycerophospholipids. There are 3 of them. One is choline, serine, and ethanolamine. So these can be ionized at a physiological pH of 7.4. So these are the structures of 3 amino alcohols. Next you have lecithin and cephalin. So these are 2 other glycerophospholipids. These are generally present in the brain and nerve tissue. So you can also find them in egg yolk, wheat germ, and yeast. So what happens here? You have the same construction, a glycerol with 2 non-polar fatty acids with a polar choline molecule attached to a phosphate group. You have cephalin. So cephalin contains again glycerol with 2 fatty acids and an ethanolamine with a phosphate group attached. So the acid that is present in both of them is palmitic acid. So both acids are palmitic acid. Now, so what is the structure and the polarity of a glycerophospholipid? So because it contains both polar and non-polar regions, it allows them to interact with both polar and non-polar substances. So because it has a polar head, it can contain the ionized ammonia, so ionized amino alcohol, and the phosphate proportion, which is going to be strongly attracted to water. You have a non-polar hydrocarbon tail, which generally is soluble only in non-polar solvents such as lipids. So this is the idea behind, this is the general construction. So when you look at the structure of a general phospholipid, this is what it looks like. And if you look at the molecule in general, and if you draw a simplified version of this, this is how it would look like. And this is the structure that you generally see on the membranes of your cells. So you have glycerophospholipid. So the venom of eastern diamondback rattlesnake and the Indian cobra contains phospholipases, which are enzymes that catalyze the hydrolysis of fatty acid on the center carbon of the glycerophospholipids in the red blood cells. So what this does is it produces a lysophospholipid which causes the breakdown of the red blood cell membranes, and once it breaks down there is no oxygen going in or coming out, so that results in eventual death. Now, so pause the video right here. So they're asking you identify each of them as a following, as a fatty acid, triacylglycerol, amino alcohol, or a phospholipid. So pause the video right here and try to solve this problem. Next, let's go to the next one, sphingomyelin. So sphingomyelin contains a sphingosine. You have a long chain amino alcohol that lipases the glycerol. Now, an amide bond is formed by the amine group on the sphingosine to the fatty acid, and a hydroxyl group that forms an ester bond with the phosphate, which forms another phosphoester bond to choline or ethanolamine. So sphingomyelins are abundant in white matter of the myelin sheath, a coat that surrounds your nerve cells. The reason it's present there is because it can increase the speed of the nerve impulses, and it can be a good insulator to protect the nerve cells. So one of the main problems with this sphingomyelins is that in multiple sclerosis, this is the main component that is lost from the myelin sheath, and because it cannot protect the neurons anymore, it deteriorates the myelin sheath starts deteriorating as the disease progresses, and you have scars that form on the neurons and impair the transmission of the nerve signals. So this is the general structure of a sphingomyelin containing a myristic acid and choline as the amino alcohol. Next we have steroids. So steroids are cholesterol, bile salts, and steroidal hormones. So you have high and low density lipoproteins that transport cholesterol between the tissues and the liver. So the A section here shows the cross section of a normal opaque artery, and the B section here is the one that shows the cross section of an artery that is completely clogged by atherosclerotic plaque. So this is completely blocked. This is a normal blood vessel. Now all steroids contain a steroid nucleus. So steroid nucleus is basically the general structure. So that consists of 3 cyclohexane rings and 1 cyclopentane ring fused together, and the rings are designated as A, B, C, D. So you have A, B, C, and D, and we number the carbons beginning in the ring A. So we start from here and you go this way. So this way we generally go from here to here, and this is how you number the structure. So let's start with cholesterol. Cholesterol is the most important and abundant steroid in the body. So it generally has a hydroxyl group on carbon 3. So this is the hydroxyl group, and it also has a double bond between carbons 5 and 6. And it has methyl groups on carbons 10 and 13. So it has methyl groups on carbon 10 and 13, and it has an alkyl chain on carbon 17. So these are the 5 main distinguishing features of cholesterol. So you can obtain it from meat, milk, and eggs, and it is generally synthesized in the liver. So it is needed for cell membranes, brain and nerve tissue, steroid hormones, and vitamin D. So it can also clog arteries when high levels are present, and it forms what we call as a plaque. Plaque is basically a buildup of this cholesterol in the blood vessels. So this is how it clogs up the entire blood vessels, and it impairs the functioning of the blood vessels. So cholesterol is considered elevated if the plasma cholesterol exceeds 200 mg per deciliter, and it generally is synthesized in the liver and obtained from foods. So a diet that is low in foods containing cholesterol and saturated fats appear to be helpful in reduction of the serum cholesterol level. So the American Institute of Cancer Research recommends that we limit our intake of foods high in cholesterol such as eggs, nuts, french fries, fatty or organ meats, cheeses, butter and coconut oil. So these are the main sources where you get cholesterol from. The highest amount that you can get is from liver of beef, which if you serve 3 ounces of it contains about 370 milligrams of cholesterol. And the least amount will be in margarine. In a tablespoon of margarine there is 0 milligrams of cholesterol. Eggs contain a large one as well. Eggs, one single large egg can contain up to 200 milligrams of cholesterol. So pause the video right here and try to solve this problem. Next, let's talk about the next steroid, which is the bile salts. So bile salts are synthesized in the liver from cholesterol and stored in the gallbladder. Now, they can have both polar and non-polar regions, so they act like soaps to make fat soluble in water. So it helps in the absorption of cholesterol in your body. And when you have large amounts of cholesterol, it accumulates in the gallbladder and you end up creating gallstones. So this is a diagram that shows the formation of gallstones in the gallbladder. So what is the general structure here? Again, the steroidal structure stays the same. You have a choline salt. There's a choline acid from here to here, and you have a glycine as the amino acid attached to the structure. And because it's a sodium salt, so we write it as sodium glycopholate, so which is the bile salt. Now there are what we call lipoproteins or lipoproteins, which are generally used for lipid transport. So lipids in general are non-polar and are made more soluble by combining them with glycerophospholipids and proteins to form water-soluble complexes. So this water-soluble complex is called as a lipoprotein. So a lipoprotein contains a fatty acid attached to a steroidal structure, which helps in the transfer of proteins from one point to the other. So what do lipoproteins do is that they surround the non-polar lipids with polar lipids and protein for the transport to the cells. And these are soluble in water because the surface consists of the polar lipids. So this is the idea behind a lipoprotein. So this is one of the reasons why viruses can enter your body as well. They can also create this lipoprotein barriers and easily enter your cell. So lipoproteins differ in density, composition and function. You have two main properties, or two main of them. One is LDLs, which is low density lipoproteins, and HDLs, which is high density lipoproteins. Now this is the composition. This is a table that shows the composition of the different lipoproteins in the blood plasma. Now lipoproteins, the mainly such as HDLs and LDLs, they transport non-polar lipids and cholesterol to cells and the liver. Now HDL is the good cholesterol, it should be greater than 60. LDL is the bad cholesterol, which should be less than 100. And the greater the value, the greater the risk of heart disease. So if the total cholesterol exceeds 240 milligrams per deciliter, there is a high chance of greater risk of heart disease. Triglycerides, if they are greater than 200 milligrams per deciliter, that's also really bad. HDL should be less than 40. If it is less than 40, then there is a risk of heart disease. And if LDL is greater than 160, there is a risk of heart disease. And if cholesterol to HDL ratio is greater than 7, that's also a greater risk of heart disease. The recommended level for cholesterol to HDL is less than 4, and the recommended level of cholesterol is 200, less than 200, triglycerides is less than 150, and HDL is greater than 60. Next, let's talk about steroidal hormones. So these are the chemical messengers that serve as communication system for the body. So these are produced from cholesterol. So you have male sex hormones, which is testosterone and androsterone. You have female sex hormones, which is estrogens and progesterone. And you also have adrenal corticosteroids from adrenal glands, which is mineral corticoids, which is used for electrolyte balance, and glucocorticoids, which regulate the glucose level. So these are the structures of different steroidal hormones. Notice that testosterone and progesterone have similar structure, but they differ in the attached group on the 17th carbon. Here in testosterone, the structure that is attached is in a hydroxyl group. On progesterone, the structure that is attached is a ketone group. Now, estradiol, so testosterone and norethindrone is also similar, but again, the extra group that is attached is a triple bonded carbon, so which changes which is generally used as a synthetic progesterone. And you have estradiol, which is generally produced in the ovaries, and which also has similar structure, but only differs the fact that the first ring is an aromatic ring. Next, let's talk about anabolic steroids. So anabolic steroids are derivatives of testosterone. So these are the illegal drugs that are used to increase the muscle mass. So the side effects include fluid retention, hair growth, and sleep disturbance and liver damage. So these are the common drugs that people take to enhance their activity. enhance their body's figure. So these are generally really bad for your body, and they are illegal for sporting events. Next, let's talk about adrenal corticosteroids. So the steroid hormones are also called adrenal corticosteroids, are generally produced in adrenal glands located on the top of each kidney, and it includes two of them. One is aldosterone and cortisone. Aldosterone is the one that regulates the electrolytes and the water balance by kidneys. Cortisone is the glucocorticoid which increases the blood sugar level and stimulates the synthesis of glycogen in the liver. So these are the structures of the molecules that you have here. So you have cortisol, which is produced in the adrenal gland. You have aldosterone, which is the mineral corticoid used to regulate the electrolyte balance in your body. You have cortisol produced in the adrenal cortex. You have prednisone, which is the synthetic corticoid that is used to replace the original structure. So pause the video right here and try to solve this problem. So with this, we end our lecture on lipids. I'll see you again in the next lecture.